We'll wait for 2 more minutes and then we'll start. Okay, so let's get started. Okay. Welcome to week 3 of Advances in Omics. The last 2 sessions we have covered a little bit of an overview of the some of the older sequencing technologies and also the current Illumina technology as well. Today we'll be looking at a few more types of sequencing technologies and finally cap it off with long read. So after that, I thought I should give you a little bit of context about genome assembly as well. Last session, I talked as an extra. I talked about the alignment parts. So alignment is when you have the reference genome and then after you generate the sequencing reads, you align the sequencing reads to the reference genome. But in a lot of cases, if you're not doing human level DNA sequencing, if you're doing of some other species of whom you do not know the reference sequence, so then you need to use assembly, genome assembly. You have to generate the whole genome just from the reads itself. So how do you do that, I'll talk about the mathematical basis for it a little bit, the algorithms that are used for that. Okay, so that would be after I discuss all the stuff that is there for this week's session. Okay, so let's get started. So first, we'll be talking about DNA nanoball sequencing. So this is a technology developed early in 2006, and it was developed by this company called Complete Genomics. And currently, I think it is owned by Beijing Genomics Institute, and it is commercialized by MGI's DNB-seq platform. So yeah. Uh, so the distinguishing factor for this particular type of technology is basically what is known as rolling circle amplification, which is basically used to create a dense packed PCR-free DNA balls for massively parallelized sequencing. So you make these DNA balls out of these reads of DNA, and you densely pack them and amplify those reads. So that is the whole purpose of DNA nanoball creation of these balls, and then amplification of these balls using a circle amplification. So the key principles include, as I was saying, the rolling cycle amplification or the rolling cycle replication, where basically you have a linearized DNA, because human DNA is linear, and you try to circularize this DNA. So basically you make this DNA into a circularized DNA strand from a linear DNA strand, and then you amplify into thousands copy without any PCR bias, and you form these nanoballs using rolling circle amplification, and the diameter is around 200 nanometers of these DNA nanoballs. Now you have patterned arrays in which DNA nanoballs are fixed in precise spots on the chip, and basically no cluster generation is happening as is happened in Illumina. So you have fixed precise spots on the chip where you have these patterned arrays, and these DNA nanoballs are kept in that. And then finally you have the sequencing chemistry, where you have fluorescent probes, you anchor oligos which bind sequentially, and then you capture the fluorescent signal, and you capture the image, and then you do base calling. So if you just had to know the USPs of, say for example, DNA nanoball sequencing, the main would be the rolling circle amplification, where you form these DNA nanoballs and you amplify it in a different manner. And the sequencing chemistry is not that far away from the chemistry that we have already studied for, say for example, for pyrosequencing or Sanger or even Illumina. Right. So I would go through the steps one by one, a little bit more in detail. So first step would be the library preparation. The library preparation is basically all the set of reads that you are going to generate. So it is the length of the sequence would be around, again, 200 to 400 base pairs. It is on the shorter range. This is around the range that Illumina can sequence at also. So around 200 to 400 base pairs of length, and then you then you ligate adapters on top of that, so that these adapters can go and bind to the oligos present in the chip, right? So two adapters are attached on the fragment ends, and these contain barcodes or sequencing anchors as well. So the first step would be to get these reads at a certain length size, and then add the adapters, which is kind of what you do for Illumina and other short read technologies as well. The next part, which is the important one, is the circularization. So the DNA fragments ligated into single-stranded DNA circles using splint oligos and ligases. So basically what they do is they make it into single strand DNA. One second. Yeah, so they the DNA fragments are ligated into single-stranded DNA circles using splint oligos. These are specific type of oligos and different types of ligases as well. And this linear DNA is digested to remove all the uncircularized molecules. So basically, you completely make all the DNA, linear DNA, into circularized DNA, and then you remove all the linear DNA that have not been converted. Then comes the important part, which is the rolling circle amplification. So there you have this enzyme from a phage. I don't think I remember exactly which phage, but this comes from a bacteriophage. So this is a 529 DNA polymerase. So the circular DNA will act as a template, and the polymerase will continue to replicate along that circle. So I don't know if you have read about plasmids and other circular type of DNA, how they are replicated. It's a similar kind of a technique here, where you have a singular circularized DNA, and then inside that you use this particular type of polymerase to make it double stranded. And it produces a long concatemer of identical copies linked end to end. So basically, it'll produce a long set of these identical circularized DNA copies which are linked end to end. So that is how you're amplifying the DNA. So you had a linear DNA, you made it circularized, then you made it double stranded using DNA polymerase, and each of these and by each circle the polymerase continuously replicates around the circle. So basically, the polymerase will amplify the DNA multiple times, and it will create a long concatenated set of DNA reads, circular, which are identical and they are linked end to end. Then all these concatemers, so these circular reads which have been amplified, they are folded into a compact structure, just like, say for example, when histone is used to fold DNA naturally within the cell. So how it does it, it has a positive charge, DNA has a negative charge, it will circularize around the histone octamer, and then it is packed densely. It's something of a similar situation here. Here obviously we don't have histone, but we have formed this circular structure using this rolling circle amplification technique, and then you compact this whole thing into a small structure which is around 200 nanometers of diameter, and then it contains 300 to 500 copies of the insert sequence. So after amplification, each read would have like around 300 to 500 copies. So the advantage to using an amplification technique like this would be that you are not using PCR. So if you are not using PCR, there is a lot of so when you do PCR, there will be a lot of PCR duplicates, there will be GC bias with the PCR as well. So those things are mitigated because you are not using PCR. And another important thing, another advantage of using nanoball is that each sorry, is anyone saying anything? Oh, okay. So what is GC bias? So basically, there are in DNA there are regions of the DNA which are highly rich in GC content. So there is a long stretch of there are across almost every chromosome there are stretches of the regions of the DNA where the GC content is very high. So regions with very high or low GC content, like with variable amount of GC content, they end up having higher or lower coverage as expected, because there is a high amount of GC rich region. So basically what happens is the DNA polymerase struggles to copy the extreme GC regions when having very high content of this GC. So the number of reads having this GC content variable, like not the exact amount of GC content that is actually present in the genome. You either have very high amount or either have low amount. So that is the GC bias. So PCR is not very good at handling regions with high or very low GC content. So basically, yeah, so it'll amplify, or it'll basically not be able to generate these amplified sequences from these high GC content regions. So basically, if you have a lot of GC, it is highly likely that the PCR amplification will not happen very smoothly. you either will have an overestimate or will have an underestimate. It is very hard to get actual amount of actual coverage of the data. So what will happen is, if you have very low amplification for these GC rich regions, the coverage in that region when you're doing the alignment will drop massively. So variant calling and those kind of things will be affected highly. So yeah, this is a drawback of PCR, but like PCR is the best known technique to amplify uh, this type of molecules, including DNA. So yeah, inherently GC bias is basically not being able to correctly estimate the amount of uh, GC, guanine and cytosine nucleotides in a stretch of a DNA, is how you would define GC bias. Okay. Yeah. So you do not have PCR, so you do not have GC bias here, but what you have is a highly concentrated amount of DNA in a single nanoball, in a single compact structure. So you have the signal density is very high, so the imaging is better, and consequently the base calling is always also going to be better. So yeah, this is the main USP of nanoball sequencing, that you create those nanoballs, and these nanoballs you are not creating with the help of PCR, so you do not have GC bias. And also this, since a lot of DNA is compacted amount in a smaller structure, the signal that you're getting is very intense. You have high intensity, and basically you have better imaging for that. Okay, so the um third step is basically loading these DNA nanoballs into patterned arrays. So you have these flow cells where you have these predefined nanowells, and each of these nanoballs goes into one of these spots, and these are separated by a certain amount of distance. So in while imaging, there is not much optical overlap between the signals coming from one DNA nanoball to the other. So you have, generally we have flow cells, but here the flow cells is basically you have patterned flow cells. So inside the flow cell you have patterns on where to keep the nanoballs, and they're separated by a small amount of distance, and it ensures that there is only one nanoballs per spot. Okay, and if you consider the density of the flow cell, it can go up to billions of nanoballs per chip. So the parallelization is massive, right? So you can parallelly sequence billions of reads of DNA together. Okay, so that would be the third step. The fourth step is actually the chemistry of how it is actually happening. So what they use is something known as a combinatorial probe anchor ligation technique, so CPAL, or it is also known as modified sequencing by ligation. So what happens is, an anchor nucleotide will be there, which will have an adapter sequence associated with it, and that complementary adapter will be present in the DNA nanoball, right? So it will bind, so the anchor oligonucleotide will bind to the actual DNA nanoball with the help of the adapter sequence. Then a set of fluorescently labeled probe will be hybridized adjacent to the anchor, and the DNA ligase will join the probe to the anchor. So the chip will be imaged to record the fluorescence. So you have the DNA nanoball, you have the oligonucleotide. The DNA nanoball has the adapters, the oligonucleotide has the adapters. They're going to be binding, and DNA ligase is going to help in that binding and joining the ends of the adapters. And then a set of fluorescently labeled probes will be used to hybridize these reads inside the nanoball, which is adjacent to the anchor, and then the fluorescence will be picked up by the imager, and finally the fluorophores will be cleaved, and the process will be repeated with a different position along the read. So basically this will you have these labeled probes, fluorescently labeled probes for particular DNA nucleotides, and for each round you are going to sequence one type of nucleotide. So in the first round, say for example, you are sequencing all the A's in the DNA nanoball. In the second, you are doing all the T's, third G and C, and so on and so forth, right? And this will generate around 150 base pairs of single end image, single end reads. And for paired ends is nothing like you have a DNA read, you sequence it from both pairs. So 5 prime to 3 prime you are going to sequence, 3 prime to 5 prime you are going to sequence. And if you do a paired end, it will go up to around two times the length, which will be around 300 to 400 base pairs, which is roughly the length also used by Illumina sequencing as well. And finally, you have these high-speed imaging, the CCD imaging modules, which capture the fluorescence for all the nanoballs simultaneously. Remember, you are having billions of these nanoballs. And then you have the base caller, which will interpret these colors and convert them into base calls. So this is basically like an overview of how this technology works. You create sorry again. Okay. How many? It's hard to say how many. So generally, like, it would be proportional to the amount of, say for example, amount of DNA in the nanoball. That would be a good estimate of that, but I am not sure about exactly how many. So yeah, I can look it up. I am not aware of exactly how many. And the length of the probe will depend from the experiment to experiment. Generally around 50 base pairs, or an oligo is generally around 50 base pairs. And sorry, yeah, around 50 to 100 base pairs is what I would assume. I'm not sure how many though. Just let me take a quick look. Genomics. Yeah, they are not mentioning the probe length. I would assume around 50 base pairs. It's a rough estimate, but yeah, you can assume around 50 base pairs. I will take a look into it. If I find any information, I'll let you know. But I'm not sure about the actual length of the probes. Yeah, this is the length of the DNA fragment and the nanoball, but not of the probe. Yeah, I'm not sure. I try to take a look, but I'm not sure if it is there or not. I'll try to find out. I'm not sure about this. Okay, should I move on, or any more questions? I hope you understood how many fluorescently, because like it'll be one at a time, like one type of nucleotide at a time. Okay, I'll move on. Okay. Okay. Yeah. As I was saying, so you have uh, yeah, you using this fluorescently labeled probes to bind with DNA nanoball and basically give out the signal with respect to the nanoballs, millions of nanoballs that will be present in the flow cell. Okay, so yeah, so the main thing to remember is here is the rolling circle amplification. This is kind of the out of the box thing that this particular technology is doing, and it is around the DNA nanoball is around 200 nanometers in diameter, and the insert sequence is around 300 to 500, and if you do paired in, the read length would be after sequencing would be around 300 base pairs. So yeah, okay. uh, Moving on is another type of technology which is using sequencing by avidity. This is kind of this is slightly a newer technique, if I'm not wrong. Yeah, so this is the paper that actually came out 2023 describing the whole technique. I've taken this image here from this paper only, so if you want to have more details, kindly follow the paper, but let us go through it. Okay, so yeah, so what will happen is you have these multivalent avidite substrates. So these are basically substrates, or you can consider them as an alternative to probes. So what was happening in probes is you had a fluorescently labeled probes, it was going and binding, and it was giving up some fluorescence, right, which was getting captured by the imaging device. Now you have these avidite substrates which are multivalent, which can attach to multiple of these different types of nucleotides, and it can give out specific signals for specific bases. So what will happen is you have these multivalent avidites, and you have these polonies, which are again kind of DNA nanoballs. So these are compacted structures of the DNA strands bound together, and inside a flow cell these DNA polonies are kept, and also these multivalent avidite substrates are added. So and then you also have to add a polymerase to kind of initiate and do the replication. So what it does is it binds to the 3' end of the primer template of a polyne and selects the correct cognate avidite via base pairing discrimination. So what is happening is you have a substrate, on top of that you have this multivalent set of kind of probes, you can assume, attached to them for different nucleotides. So what will happen is when you have this DNA polynese, you have the DNA polymerase. The polymerase, when it will replicate the polyne, it will come and pick up the actual avidite required for this particular base pairing. So the multivalent avidite interacts with multiple polymerases on one polyne to create avidity binding that reduces the effective KD, which is the dissociation constant of the avidite substrate 100-fold compared to a monovalent labeled dinucleotide. So what is happening is these signals are much more concentrated. These are bound very hard together. So, as you were saying, that the effective KD is reduced. So basically the dissociation constant is reduced, so it is more bound tightly, allowing for a productive binding of nanomolar concentration. So even if at nanomolar concentration, the binding is very strong. And multiple polymerases mediated binding events per avidite ensure a long signal persistence time as well. So you have this because you have multiple of these so in the same stretch of the DNA you'll have you have ATGC, right? So you have multiple of these avidite substrates coming and binding to this polonies, right? So yeah, this also ensures a long consistent signal persistence time as well. And this again, after this these substrates will generate fluorescence, which again will be captured by the imager, and then base detection will be followed after that. And following the detection, avidites are removed from the polonies. Extension by one base using an engineered polymerase incorporates an unlabeled block nucleotide, and then a terminal 3' hydroxyl is regenerated on the strand, allowing the repetition of the circle. So at one step you capture all possible kinds of uh, nucleotides that are present for one step, and after that is done, you remove all these avidites, and then you do the second step for the second set of nucleotides, right? So generally this generates the USP of this is this creates this very tightly structured kind of DNA nanoball structure, which are known as DNA polonies, and the signal intensity is very concentrated in a particular position. It is not since these are tightly kept together, they are not scattered around. So that is one of the major USPs for sequencing by avidity. So uh, so this is actually the first step. So you have these avidite molecules, you have the DNA nanoballs or polonies, and each of these avidite molecules will go and bind with this, and each type of polony will give you a type each type of these avidite molecules will give you a type of base pair, base that is getting detected, and then you wash off uh, these avidites, and then you basically you have to reversively terminate the nucleotide so that the blocking is removed, and the next step of polymerase can act on it and start sequencing again. So this is the first deck is kind of showing you the repetitive procedure. Now this is a more of a the second row is giving you a more detailed image of how this polony stay together, and the avidites interact with it. So this is how a single avidite bound to a DNA polymer is kind of that's the kind of schematic for that. The initial surface primer used for the library hybridization and extension during the polymerase is shown in blue. So this is the actual primer used on which the DNA polymerase will act. Sequencing primers are annealed to the single-stranded DNA polymer, because again you are using DNA polymerase, you need these single-stranded primers as well. And after that, each arm of the avidite, which is the black, these black arms, each arm of the avidite connects the avidite core containing multiple fluorophores to a nucleotide substrate, and the polymerase bound to the sequencing primer selects the correct nucleotide to base pair with the templating base, which will be the set of reads that are there in the polymer. The result is multiple base mediated anchors points non-covalently attaching the avidite to the DNA polyny, and then you use this multiple DNA polonies with template specific avids bound during this binding step of the cycle, and you can image these using whatever technique you use for imaging, and then you do the base calling basically. So again, just a quick recap. For DNA sequencing by avidity, the USP is that you have these multivalent avidite molecules which contain fluorescent signals for all the different types of nucleotides. You use them on DNA polonies, which are kind of small set of nanoballs of DNA. Then you use a similar type of a technique where you have a DNA polymerase, you have a primer, and instead of having like fluorescently labeled nucleotides in the solution, you use these avidites to kind of to do the synthesis basically. So you do not have free floating DNTP or anything else. You just use these avidites to do the synthesis, and then based on the the signals from these avidites, you generate what the base is there at that particular position, right? So this was the USP for avidity. The USP for nanoball sequencing is the rolling circle amplification with no PCR. So these are more niche sequencing technologies which are not so commonly used. These are used for very specific tasks. These are like not so commonly used like Illumina or a Sanger or something like that. These are specific uh, use cases where these are used. So yeah, okay, okay. Yeah, so I think I've given yeah, I've mentioned this paper. You can get a read on this paper. Try to see if you have any uh, uh, interest in technologies like this. One thing I would want to highlight is, see, the the if you have these homopolymers, we have discussed in earlier sessions as well, the homopolymer length is detection is a big issue across different technologies, because you have the same base pair repeated multiple times. So what happens is the signal peak does not come down, it stays at the peak, and the intensity of this peak will be proportional to the number of these homopolymers in that particular region. So for so the NextSeq and the NovaSeq, these are Illumina technologies. So you can see the mismatch percentage is quite higher across homopolymer regions, while avidity or sequence by avidity, here the homopolymer mismatch rate is quite low. Okay, so this is also one of the um, USPs of this particular technology, that you can actually get a decent amount good accuracy for the homopolymer lengths for using sequencing by avidity compared to a Illumina or something like that. Yeah, okay. So yeah, uh, you can take a look at this paper. This is actually very decently written paper where you can understand what is happening. Okay, so moving on, the next type of technology we'll be talking about is sequencing by binding, or also yeah, sequencing by binding. PacBio actually uses sequencing by binding, not sequencing by synthesis. So anyway, so the thing is, sequencing by binding, the USP is that it is very accurate. This is, and Q40, you remember the quality of the base. So this is a Fred scale. So Q40 meaning 1 in 10,000 bases is the error rate. So that's pretty low and quite good. So how it works is, basically you have a binding phase, a detection phase, and an incorporation phase. So at the binding phase, native nucleotides are presented with polymerases, forming stable, non-incorporative ternary complex with the template. So uh, yeah, so and this binding depends on correct base pairing. So you have a set of native nucleotides, and they are presented with a polymerase, and they form this ternary complex with the actual template, and upon binding, a fluorescent signal will be generated and captured, and this will identify the base of the signal without chemical incorporation. And after imaging, the nucleotide is incorporated, regenerating the 3' hydroxyl for the next cycle. So what you're doing is, you are binding native nucleotides with the template, and then you're capturing the signal, and then you're removing the native nucleotide, and then the unlabeled nucleotide will be incorporated, and this uh, process will go on, right? So yeah, just give me a moment. I'll be back. Yeah, sorry for that. Okay, so yeah, sequencing by binding. So how is it actually different from sequencing by synthesis? Here, what you are doing is you are incorporating a stable uh, ternary complex uh, to detect what base is there in the uh, in that particular location, right? And you are doing it reversibly. So you have this ternary complex, you are incorporating them with respect to the template with the help of the polymerase, and you're capturing what signal is coming out of there. But that cannot be continued for the next base. You have to remove that ternary complex, get a actual native nucleotide, and then continue. So sequencing by binding, what it does is it decouples. Same with avidity binding as well. So there also the concept is similar. You are decoupling the steps of uh, the DNA synthesis and the detection of the base that is there. In sequencing by synthesis, along with the synthesis, you are detecting the base. In case of sequencing by binding or by avidity, what you are doing is you are using an external complex to come and bind at that location. You're using a imaging system to detect what base is there, and then you're removing that, and then you are adding a normal nucleotide, and then you're doing the next step. So that is the major difference, is basically you are decoupling these steps, which leads to more accuracy. That is very true. You will see much more accuracy in binding and also by avidity than by sequencing by synthesis. So these are having extreme accuracy, and uh, they also use native nucleotides to avoid chemical modifications. There we are not using native nucleotides, we are using some fluorescently labeled nucleotides, we are using some radioactively labeled nucleotides, or some modified nucleotides, or DDNTPs or something like that. Here you're doing native nucleotide incorporations, but before that you are incorporating a complex ternary complex from which you can determine what base was there. But then after while doing the synthesis, you're using normal nucleotides. So the incorporation is also very seamless. And obviously, if you have like uh, if you are doing something that requires very high precision, sequencing by binding is a better technique to use compared to a sequencing by synthesis technique. Okay, so Illumina, I'm not going to be discussing in great detail. I hope you remember the first class only. I think if I remember correctly, first class only. I showed you one video for Illumina sequencing and how actually it occurs, the clonal amplification and all the sequencing by synthesis and so on and so forth. I'm going to link that video here. If let me just go. Illumina. Yeah, this actually I like this video very much. This explains very well. So yeah, we have discussed this in the first session only, but yeah, just let me give you a quick recap. You have these DNA reads. So you have these DNA strands. They are going to go and bind in the flow cell. There are millions of oligos attached to the flow cell. So these set of reads are going to go again having adapter sequences, and also the oligos will have adapter sequences. So these are going to go and bind to the oligos. Then DNA polymerase is going to come and make double strands, and then like the double strand will be will be cleaved and made into single strands, and then these single strands will be bound to the next set of oligos attached to one. So it forms a bridge kind of a structure, and this bridge is amplified using DNA polymerase, where you do and this technique is known as bridge amplification and clonal amplification, and which is one of the like kind of one of the USPs of Illumina, right? But there are some issues with Illumina as well. The first issue is with respect to the size of the fragments. Illumina, as I have mentioned earlier, also is a short read sequencing technique. It can give you around 300 to 500 base pairs depending on your experimental procedure. But this is a very big limitation when you are trying to so you remember about the alignment that I talked about last session. So if there are mismatches in the alignment in with respect, so your reads and your reference, if there are mismatches of base pairs in your reads with respect to the reference, you can call that there is a mutation probably there in your sample compared to the reference one, right? So if it is a single base pair mutation or a very small set of base pairs while a mutation, a small insertion or a small deletion, then it's fine. A 300 base pair length read can detect that. But there are also larger variations that occur in the genome, right? So you have these bigger structural variants, we call it, variants that are bigger than 50 base pair, and these can go up to like in MBs, in megabases. Like, so generally you have you have large sections of the genome deleted, uh, translocated from one region to the another. You have inversions of sequences. You have a lot of these complex big rearrangements in the genome, which can be benign, which can be pathogenic depending on the context. But detecting those bigger uh, bigger rearrangements in the genome is very difficult when you have reads which are not spanning the whole event. So for example, you have a deletion of, say for example, 2 kb 2000 base pairs deletion in a particular region of the genome. So if your base is only 300 base pairs, it is not going to cover the whole deletion event. But if you imagine, if your length of the read is around 5 kb or 10 kb, right, 10,000 base pairs, inside the 10,000 base pair you can very clearly visualize the 2000 base pair deletion, right? But if your read length is smaller, detecting larger variations becomes a very big issue. You can detect, it's not that you can't, but that comes always with some caveats and some uh, error rate, very high error rates, and it always cannot detect the breakpoints actually where the deletion or the insertion is happening. It is very hard to detect using short read data. So that is the major limitation of Illumina sequencing, is the length of the read, right? So because the speed and the accuracy is good enough, speed is quite good, is the issue is the length of the read. The Illumina sequencing can take a long time to generate the sequence data, but like that is also true for other sequencing technologies as well. Illumina is not that bad. It is bad compared to some others, but it is not bad, like that bad that you cannot. The time is the main drawback of it. It's not that case. And also another downside would be that there is no real-time access to the data, and you have to wait for the whole experiment procedure to finish to actually get the data, right? You cannot get it like real time. So that is another issue, but the main issue is the length of the read, right? So there comes some different sequencing technologies which kind of addresses these drawbacks. The first would be SMRT, which is single molecule sequencing. So it is it is also based by sequencing by synthesis approach. So what happens is the DNA is synthesized in some small well-like structures. So these are known as zero mode waveguides, or ZMWs. These are small well-like containers uh, with the capturing tools located at the bottom of the well. So inside the flow cell there are thousands of these wells, very micro wells. Inside the well you have these wells are known as zero mode waveguides. We'll come to that later. Why? And at the bottom of the tool, not at the top, the bottom of these wells, the capturing machines or the imaging uh, machine is kept. So the image or the signal is gotten from the below, not from the top, right? So the sequencing is performed with the use of unmodified polymerase and fluorescently labeled nucleotides flowing freely in the solution in those microwells. These wells are constructed in a way that only the fluorescence occurring by the bottom of the well is detected, and the fluorescent label is detached from the nucleotide at its incorporation from the DNA strand, leaving an unmodified DNA strand. So uh, yeah, so this technique can go up to 1000 base pairs. So it is slightly better than actually Illumina will go up to 300 to 500. This is around 1000. Okay, so what happens is you have these inside the well. So this is happening inside those ZMWs, right? So inside the well you have the your actual read. You will be having a primer, and then you will be having the DNA polymerase. So the DNA polymerase will pick up these fluorescently labeled nucleotides, and it'll keep on adding one by one, just like sequencing by synthesis. And the USP here is that the signal capturing technique is slightly different. So it is captured from the bottom of the well, and it is read at each of these of the rounds of adding of these nucleotides. So it is very similar to Illumina in terms of uh, the sequencing by synthesis procedure. The detection of the signal is and the amplification techniques and all those things are different with respect to SMRT compared to Illumina. And because of this the detection technique, this can go up to 1000 nucleotide, a slightly better version of Illumina. But this actually like you have these thousands of these wells inside the flow cell, but still the throughput of this is slightly lower than that of Illumina. So Illumina will be a little bit more efficient in terms of uh, getting much more number of reads sequenced in a similar amount of time. So this will be the throughput will be slightly lower compared to Illumina, but the read length is higher. So that's a kind of a trade-off that you need to address. Okay. Okay, so now we come to the actual long read sequencing technology. So SMRT, the technique is very good, but it only is getting you up to 1000 base pair. So we need more than that. So here comes PacBio hi-fi reads. So these are so the way of producing these reads is slightly bit different compared to other sequencing by synthesis technologies. So what you make is a circular consensus sequencing mode used by these PacBio long read systems, and what you make are known as hi-fi reads. So this is a proprietary name used by PacBio. It does not mean anything per se, but it's just that how they are making these reads, circularized reads, they have named that hi-fi reads. It's nothing more than that. But these reads are, if you remember, where we also saw circular reads. If you just go back here, yeah, DNA nanowall sequencing. We were using circular reads. We were having single-stranded DNA. We were making it circular. We were amplifying it, and we were saying that this is a good idea in terms of the accuracy, and also the rolling circle amplification is a good thing compared to the other PCR amplification and so on. So the same concept applies here, except for we are using SMRT technique, not the nanowall technique, but the kind of reads that we are generating is similar. We are having these are the single linearized DNA. If you break it down into single strand, and then if you attached end to end, and if you use polymerase on the inside to make it double stranded, it will make this circularized DNA, right? So these hi-fi reads can be used across a wide variety of SMRT sequencing applications, from whole genome sequencing to de novo assembly, variant detection, and so on and so forth. So what happens is you have these linearized DNA, then what you add is this rounded bell library. So basically, at the end of the two reads, you have these shapes where a small portion of it is double stranded. So this portion is double stranded, but the rest of it is circular because there is no complementarity in the sequence. And so what happens is, then inside this thing you are going to add these primers, and then you add the polymerase, and then basically rip off the middle portion here, and then it becomes a circular DNA, right? So the circularized DNA is sequenced, repeated in passes, and the polymerase reads are trimmed of adapters to yield subreads, and the consensus and the methylation status are, we are not going to go into that details, but using these kind of circularized DNA helps you get rid of PCR bias, and also is good for very high accuracy of the reads as well. And this PacBio has its own technology using SMRT, where using this circularized reads and this hi-fi reads, what they call, they can go up very high. So this can go up to say 10 kb to 20 kb, even higher. This is slightly lower than the ONT data that we see, Oxford Nanopore, that is also long read, but this is slightly lower. The read length is slightly lower than that, but the accuracy of PacBio is almost the best in across long read technologies. ONT has a lot of errors, but PacBio is very good with respect to accuracy, and also it is very good in terms of the length of the read. So this is one of the widely used technologies today with respect to long read data. But again, I'm going to point it out. In real life, PacBio is much more expensive compared to ONT. So the use of ONT is much higher compared to PacBio because of the cost of the technology. Yeah, coming to ONT, this concept is I think the technique that is used is quite different to what we have seen till now. Sequencing by synthesis, sequencing by avidity, using circular reads, and so on and so forth. We have seen a lot of techniques, but this is kind of quite different from what we have seen, because the signal detection that we are doing is with respect to the current passing, and not with respect to some sorry, I missed this comment. Yeah, that is true. Yes, because you have these reversible transcriptions. The thing is, the best way to do this sequencing would be, as I was saying, that you decouple the detection part and the synthesis part. If you can decouple that, then there will be no scars left. But since Illumina is not decoupled, during the synthesis only you are detecting. So yeah, it leaves scars in the template also, and it can cause errors while sequencing. So yeah, it's always better to decouple, but yeah, you have to have a balance of everything, right? So there needs to be a balance of the throughput, there needs to be a balance of the length of the read, there needs to be a balance of the cost as well. It cannot be very expensive so that people cannot use. So all the technologies will have drawbacks, and the drawbacks are there with respect to all these things that I mentioned. Either it will be cheap, but it will not give you longer reads, or it will be very expensive, it will give you longer reads but will not give you the accuracy. So there will be trade-offs all over the place, but yeah, you have to choose what question you are trying to answer. Based on that, the technology will be used. So as I was saying, if you're wanting to detect these larger variations, then using Illumina is not a very good idea, because the reads will be shorter, you need longer reads than that, right? So ONT or PacBio would be useful there. Now if you do not want to sequence the whole genome, you want a targeted sequence, and you want to detect specific mutations, single nucleotide mutations there, so there the accuracy is much more important than the read, right? Which in those cases you will use short read technologies like Illumina. Illumina will give you very high accuracy compared to a longer read technology like a PacBio or a ONT. So yeah, you have to choose based on what type of question you're trying to answer from the data. Yeah, that's correct. Okay. Yeah, so as I was saying, ONT produces very long amount of reads. Here, what happens is this is a very interesting technique here. So you have a double-stranded DNA. What you do is you make it single-stranded, and then you have a very complicated kind of a protein here. But this protein is very similar to the actual protein present in your mitochondrial membrane, which is the F0 F1 ATP synthase, or the actual hydrogen pump used in the electron transport chain, which actually makes the ATP, right? So this enzyme is kind of very similar to that. There also there are two modules, and one module moves with respect to the other. Here also it's a similar kind of a protein. So this is a membrane protein, by the way, right? So this is a microscopic pore in a membrane, and then there is a protein, and inside through that protein the reads are passing, right? So or basically, yeah, the reads are passing. So the two set of modules on the protein, they have different functions. So on the top, the protein will unzip the DNA helix into two strands, so it will make it single stranded, and one of the strand will go inside through to the next protein, and the next protein will create a pore in the membrane and will hold an adapter molecule. So this is the adapter molecule. This read is going and binding here, and it makes the read stable here, and current basically flows, or a flow of ions happens, and you measure the current. So a flow of ions through the pore creates a current. Each base blocks the flow to a different degree, altering the current, and then the adapter molecule keeps the places in long enough, and based on the difference in current, you tell what base is there, right? So earlier, when we were using fluorescence or radioactivity or things like that, each of the base had a specific signature to it. So if C is getting added, there will be a particular fluorescence. If T is getting added, there will be a particular radioactive signal, so on. So each had a different signal there, right? So here also the signal is basically the change in the current, the amplitude of the current, and based on how much of it has changed, you can predict what base was there. Okay. Yeah. The image is not very good, but I'm just going to go through the step. So basically, the nanopore is inserted into an electrically resistant membrane. So the protein complex, a potential is applied across the membrane, resulting in a flow of current only through the aperture at the nanopore. The single molecules enter the nanopore, which will be the read bases. It will cause characteristic disruptions in the current, and by measuring the disruption, the molecule can be identified. Right. So the signal processing part is different, because here we are using the signal used is quite different to the other technologies. Yeah. The issue is, again, one issue I'm going to point out is the homopolymer issue. If you have a like here, probably it's not very visible. Here you have 4 T's. The T's are like marked well. The signal has come out well, but in most cases, what will happen? It'll have a flat peak. It will not have a peak and a trough. It will have a flat peak, and detecting the number of bases, repeat bases in that particular region will become very difficult. So Oxford nanopore technology has some drawbacks. It gives you the advantage of it is it will give you a very long read, and also one more thing that you can detect while doing this is the methylation status, right? So for example, if you have a here, you have a modified base on top of that, say a 5MC mark or a 5HMC mark on that particular base, modified base. So you can also detect the epigenetic modifications on the base directly from this experiment only. You do not need to do a separate sulfide binding experiment or some other experiment to tell at what bases, at what points of the bases are they are modified epigenetically. Here you can detect the base, and also if there is a modification on the base directly from the same data. So that is a very good advantage of ONT. The issue is, and the other advantage is the very high long reads. The issue is the accuracy. Uh, Homopolymer errors are very quite common. I have seen it firsthand. It's very very common. If I get time, I'll show you one example of such. Okay, let me show that. I think that would be more interesting. Just give me a moment. I hope you can see my screen. Just let me enlarge this thing. Okay. Now, I don't know how many of you have seen IGV before. So, the tool is known as IGV. It's basically a genome browser, genome viewer. So, there are two panels here. This is one panel, and this is the second panel. And in below, this is actually the reference, right? What are the base sequences in the reference? The ones in below are basically codons. So CAG will code for Q, GGG will code for G, and so on and so forth. And on the bottom, the blue one is actually the exon of the gene. So what is happening, if you see clearly, the top panel is from short read exome data, and the bottom panel is from long read ONT data from the same sample. Right, you can see at this, the one that is marked here, you can see there is a gap in the reads after alignment. So this image is after alignment, right? So you can see there is a gap here. In some reads, the gap is 2 base pairs. In most of the other reads, the gap is 1 base pair. In the same sample with short read data, we did not find any indel here. Now, actually, if you look into the sequence of the bases in the reference, you see a homopolymer region here. There are 5 Gs here. So why? So clearly this is an error, right? Because in short read, it has more accuracy. It has not detected the indel, but in long read, with lower accuracy, it has detected an indel. So definitely the indel is an error. But why is the error happening? If you see, there are 5 Gs here. What the ONT data has done is it has counted 4 Gs. So it has assumed 1 G has been deleted here, which is actually not the case. So this happens. So if you do variant calling on this data, I have also done variant calling on this data. So I found an indel here, and this was actually a pathogenic variant. So I thought probably this is actually these are samples of rare diseases, of individuals having rare diseases. So if you see a variant like this in the coding region of a gene within an exon of a very important gene, you can assume that this variant will cause something, at least some disruption. If an indel like this is happening, this will cause the frame to shift, protein sequences and all those things will be affected, right? But that is not the case, because if you look at the short read data, this data is much higher accuracy. There is no indel here, but here you can see the indel, and that indel is spurious because you have a homopolymer. It's not a very big homopolymer. You can see these are 5 Gs, nothing more than that. These are not like 10, 12, 15 base pair long homopolymers. These are 5 Gs homopolymers. The base caller has read 4 Gs here. So that is why it has introduced an indel that 1 G is missing and 1 G has been deleted. But this is a spurious call. Another example I have is a very similar one. Here it is kind of the orientation is opposite here. Here the bottom panel is the short read data and the top panel is the long read data. Right? In long read data, again, you can see there is an indel, one base pair deleted. Here again, there is a homopolymer. 4 Cs, the ONT data has counted 3. And that it has introduced an indel here saying that 1 C is missing. But this is a spurious call. If you see at the short read level, there is no such indel here. The reads might be longer, but the accuracy, there is a trade-off. This is the trade-off I was talking about. The accuracy will be hampered very badly, right? But again, if you, I don't have anything handy here, but if I get the time and the opportunity, I will show you the advantage of long read. So if you have long read, how are bigger variants detected much better than the smaller variants? So yeah, I'll probably cover it. If I have anything, I'll cover it in the next class. But these are actually practical problems that we face using with respect to homopolymers, with respect to long read and short read. So the bottom panel for both of them is basically a short read Illumina sequencing data. This is a targeted sequencing data. You can see that coverage is dropping off because this is only targeted towards the exonic region. So this is exome data, but for long read, it's whole genome data. You have the whole genome, so the coverage is not dropping off. Here the coverage is dropping. So here actually the exon is ending, and after that you can see the coverage is dropping off. There are not much reads here, and so on and so forth. So even then, it has much better accuracy than long read at smaller length base pair resolution. Okay, so yeah, mostly that is it in terms of what I wanted to discuss. Yeah, pros and cons also, like I think I have discussed like the major cons and the pros. Yeah, so any questions from here? If not, I'll be moving to the assembly part a little bit. Okay, so let me quickly cover the assembly part. So I'll be talking about what genome assembly is, what is coverage, how actually we do assembly. So all this example that I was showing, the sequencing part, this one, the reads are aligning to a known reference genome. So we know that this is the sequence here, the reads are coming and aligning here. But in cases where you do not have the reference genome, you have to generate the reference genome from these reads as only. How do you do that? One good thing for while designing the reads is we do overlaps of the reads. So one read will overlap with the other read will overlap with the other, and like that we can generate end to end. It's a very simplistic view of doing, but that is the general idea. And walk through the mathematical basis for that. I'm not going to go into very much detail, but if you have any questions, definitely stop me and ask, or we can also have a discussion later as well. Okay, so yeah, there's a very close link with respect to coverage and assembly, right? Coverage is basically at one position, how many reads are covering that particular base pair, right? So again, let me go back to this example. There is a G here, right? Let us forget the indel, wrong indel. There is a G here. How many reads are covering the G? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Around 17, 18 reads are covering the G position here, right? So if you do an average across the genome, across every base pair, what is the average number of reads covering that base pair? That is what coverage is, right? So on an average, a single base pair in the genome, how many reads are covering that? So that is by definition what is coverage. And the basic principle of assembly is that the more similarity is there between end to end. Why do we have coverage, so high coverage, 20, 30 coverage like that? Because the reads are overlapping with each other, right? So in assembly, the idea is the more overlap that you have, the more better assembly you are going to get, and the more overlap that you have, more likely that they have originated from the same overlapping stretches of the genome. So that is the assumption that we go through, right? So say two reads truly originate from overlapping stretches of the genome. Why might there be differences? So there can be two things. One is there is a sequencing error, or there has been a difference with respect to the inherited copies of the chromosome. So humans are diploid, we inherit one copy from the mother, one copy from the father, and these copies might not be exactly similar. There can be differences in base pairs there. So this is a very good example for that. This is a mother sequence, this is a father sequence, and combined both of them is the son or the daughter sequence, right? So in that case, in one strand there is a G, one strand there is a A. So these are basically these can be either mutations or sequencing errors depending on the evidence across both, right? So how do you find these overlaps between the reads? So and how do you represent them? So the data structure that is used to represent them in mathematical form, also in computer, when you are coding this program, is use of graphs, right? So graphs are basically data structures which have nodes which are connected to each other by edges. So if two nodes are similar, they will be connected, and there are a lot of caveats of graphs. I'm not going to go into that, but the idea is that if you have in this case, if you have two nodes representing two different reads, and if there is an overlap between them, you have an edge between them. So that is how generally how graphs data structure would work in case of assembly, right? So yeah, so you build a directed graph, right? So sequence A overlaps the suffix of sequence A overlaps the prefix of sequence B. The suffix of sequence B overlaps the prefix of sequence C, right? So there is a order. So sequence A to sequence B to sequence C. So this order is also maintained using directed graphs. So the edge originating from sequence A to sequence B, the arrow will come from sequence A towards sequence B, and from sequence B towards sequence C, right? So there will the arrow, the edge will have a direction, right? So these are known as directed graphs, right? So generally that is how you make the graph. And the more overlap there is, so what you are trying to do is finding the suffix of read 1 and finding the prefix of read 2 and seeing how much overlap there is, right? So the more overlap there is, the more higher weight of the connection, the edge will have a higher weight, right? So yeah, so this is an explanation of what a graph is basically, as I was saying. So you'll have these vertices, which are also known as nodes, which are A, B, C, and D, and these will be connected by the edges, or basically the connections, what we say, the edges. So the edges will be, so this is a mathematical representation of a graph, right? So this is how you represent. You give V equal to how many nodes, what are the nodes, and through E you denote what are the connections. So A and B are connected, so you put it in first brackets. A and C are connected, you put it in first bracket, and so on and so forth. D is not connected, so D will not be present inside the edge set, right? So this is how a directed graph mathematically looks like, and from overlaps we know we can make each of the nodes. So each of these A, B, C, and D can be each of these reads, and depending how much overlap there is, you can draw an edge from A to B or A to C or whatever, right? So an overlap graph, as I was saying, will be constructed like this. So sequence A, sequence B, and sequence C. So what you do is overlap this suffix of sequence A with the prefix of sequence B, and suffix of sequence B with prefix of sequence C. You can also do C to A as well, right? So this is how an overlap graph will look like. So the weight of each of these labels, so you can see the edges are there, and on top of that the number is written. This number corresponds to the number of bases that are overlapping. So yeah, so this is how graph will look like. It will become circular if your DNA is circular, or if your DNA is circular, it is definitely going to become circular. It is also going to become circular if there are repeats in the reads. If the same repeat units is present across multiple reads, there will be overlaps, and there will be this circular overlaps, because A will be overlapping with B, B will be overlapping with C, C will also be overlapping with A, because all these three reads have a similar type of a repeat sequence, right? So generally you get the circular graphs inside the whole big graph. You'll get the small modules of circular graphs because there are repeat units. And also, if your DNA you are sequencing is a plasmid or some sort of circular DNA, you're going to get a overall circular graph. right? And yeah, so how do you find the graph, find overlaps? I'm not going to go into details, but the idea is that uh, you have so if you're finding an overlap of x and y, what you're trying to find is the overlap of the suffix with the prefix of y, suffix of x with the prefix of y, right? So say for example you are assuming the length of overlap would be something around 3. So what you do is you try to find the suffix in the prefix of this overlap of the next sequence. So x has GCC here, y has GCC here. So if you do an overlap, the first 3 elements are overlapping here, then you move on to the next 3 elements GAT, and then you also see there is a GAT overlap here. So this, if you combine these two sequences, what will be the sequence, right? CTC TAG GCC, that is common between the two reads, and then again CTC. So combining both the read, you got a end-to-end sequence of the genome. So it started from the CTC, which is not overlapping with the next sequence. Then there is a 6 base pair overlap of x with suffix of x with the prefix of y. That is the common sequence, and then you have another sequence which is not overlapping. So like this, if you do it for multiple hundreds of reads, you can get a decent amount of DNA sequence directly from here. Okay, so this is how a graph will look of an original. So this is the original string. What they have done is basically gotten all the suffixes of this particular string of a certain length. I think here the length is 1 2 3 4 5 6 7, so 7 base pair length, and it is doing 3 base pair overlap. So the read length across all of them will be 7 base pairs. So you'll find all the 7 base pair combination of the sequences from the original string. Then you are going to do overlay of 3 base pairs, minimum of 3. So if there is a minimum of 3 base pair, you're going to connect. So for example, this to this, the CGC suffix overlaps with the CGC prefix here. So there is a connection of 3 here. Also CGC overlaps with CGC here. So there is a connection of 3 here. Here there are more connections, 6 and 5 and so on and so forth. So all these connections will make you a graph like this. So you can imagine a sequence of length, I'll assume this is like around 30 base pairs, if you assume, will give you such a complicated graph. Imagine the complicated how complicated a graph would be if the length of the original string is 3 billion base pairs. So we have different computational techniques to handle that. I'm not going to go into the computational details, but the idea is to find the shortest common. So if I formalize all these things, finding overlaps and using graphs and so on and so forth. So if I want to summarize this, what you're trying to find is basically a shortest common substring between two sets of reads. That is the computational problem that we are trying to solve, right? Okay. Yeah, I'm not going to go into detail, but one thing is, yeah. So if a modified overlap graph where, so the idea is that how are you going to get the whole sequence? Say for example from here, how are you going to get the whole sequence? You start from a node, then you move to the next one, then from here you either move here or here, and you take all the possible combinations of the roots, and then you find, after traversing the roots, you find the set of sequences, right? So the issue, yeah, anybody wanted to say something? Okay, I'll continue. Please let me know if you have any questions, right? So if you have say for example, a modified overlap graph where each edge has a cost. So if you travel through that edge, because there will be multiple options, right? From one node to another, there will be multiple ways to travel, right? So you have to put an algorithm where the edge will have a cost which is proportional to the negative length of the overlap. So the shortest common substring corresponds to a path that visits every node once, minimizing the total cost along the path. So that would be the algorithm. Because there will be multiple paths, what you are going to do is you develop a scoring system where according to the path that you travel, you are going to get a score. And once you have all possible scores, you choose the one which has the minimum score, right? So that would be your best overlap. But if you check this problem out, this is known as a traveling salesman problem, and this is NP hard, meaning this cannot be solved in finite time by the computer, right? So there are this problem is not solvable. So what we do is an approximation of this problem, which is known as the Hamiltonian path problem, which is NP complete, and yeah, we can move from there. So we kind of dumb down the algorithm a little bit, so that if you want to consider every possibilities, the we will never end the algorithm, right? So we need to do yeah, different modifications to that. Yeah, I'm not going to go through all this, but these slides will be there. If you want to take a look, please take a look. If you have any questions, please, I have given my email ID, please write me an email or ask in class, whatever. Yeah. Yeah, so generally the idea is for assembly is you have these overlapping reads, you find this overlap, you find the layout of the graph into contigs, and then you pick the most likely nucleotide sequence for each contig, meaning the best path for that, right? And yeah, so these are different specialized data structures. We have discussed about suffix trees in the earlier class, but suffix trees can also be used to find these overlaps and create the graphs, right? So the idea is that the basic idea is that you build the tree or build the graph, and for each string you walk down from the root and report any outgoing edge labeled with a separator, and each path will correspond to a prefix or a suffix match involving a prefix of a query string and the suffix of a string ending in the separator. So that is the basic idea. You start with a node, you have multiple paths to walk, you walk all the nodes, you have some scoring system, and you get the least one. Okay, and these are other examples. I'm not going to go into this. Yeah, one very widely used technique is using dynamic programming. I think I'll touch upon a little bit of dynamic programming in later classes about for alignment or something, some stuff, other stuff. So yeah, so generally the number of overlaps, if you do just by greedy algorithms or brute force, yeah, the number of overlaps would be around O(n) square, and then size of each of the dynamic programming matrix would be around OD square. So overall it will be basically very high, square, right? But if you do it with a suffix tree, it becomes linear. So the time basically scales down by a lot. So the general idea is not different. What we are trying to do is we are trying to optimize the algorithm as much as possible, because two things: the length of the reads are very small compared to the length of the sequence, and the overall number of reads are very high compared to the length of the sequence again, right? So for both of these, we use different algorithms and different optimization techniques of these algorithms. So this is the final thing that I'm going to be discussing, is something known as a De Bruijn graph. It is also somewhat of a graph. So what you do is you define a k-mer. K-mer is basically, the k stands for the number of elements in that string. So in this case, these are all 3 mers, because these are made of 3 base pairs. So you make a k-mer of a substring of a length k, and then you make all those k-mers there. So basically you get all these k-mers, and then you make it into a graph, right? So you make a graph like this, and depending on the overlaps, you make a graph, and this is known as a De Bruijn graph, right? So I am not showing so if you make a De Bruijn graph out of the whole human genome, this is going to be a very big graph, and this is a directed graph based on the overlaps. And how you make it based on the k-mers. So k-mers will be determined by how efficiently you want to run your algorithm. If you want your algorithm to prioritize accuracy over speed, you go with a smaller k-mer. If you want to do it speed over accuracy, you go with a larger k-mer, larger overlaps. So the k length will be the amount of overlap between 2 reads, right? So that is how it's done. Now once you have made the graph, how do you infer the graph? How do you get the whole sequence end to end, right? So there's something called random walk. So on graphs, you start from one node and you follow the edges and you walk on the graph, right? So moving from one node to another node is known as a walk, and the random if you walk it in a random sequence, it's a random walk, right? So this is basically a random what a random walk looks like. So these are all probabilities. So you start from node 1, right? This is your first step, first iteration. In the next iteration you have 50-50 chance of going to node 2 and node 3, right? So this is 0.5, this is 0.5. Now from here, node 3, in the next step, you can move to node 1 or node 4 or node 5. So these are the probabilities of those. So you calculate the probability of moving from one node to another. Now this is a graph which does not have edge weights. In our our De Bruijn graph, it will have edge weights, right? So those weights are considered while you are doing calculation for this probability. And ultimately what we do is known as an Eulerian walk. So this is how a De Bruijn graph looks like. So this is the string, and for an example, this is how a De Bruijn graph looks like, and you do a Eulerian walk on this. So the Eulerian walk is basically the idea that you can visit each edge at exactly once, not more than that. So for example, if you're moving so if you're moving from this to this, you can use this edge first, and if you are moving it the second time, you have to use this edge, the second edge, right? If you do not do that, that would not be an Eulerian walk. So Eulerian walk is a special type of random walk with some constraints on that, right? So so the idea of genome assembly is you break down the bigger reads into smaller reads, you make k-mers out of those reads, and you build a whole De Bruijn graph. Like it's a graph of those reads, and you connect those reads by the edges based on their overlaps, prefix and suffix overlaps. And then how do you get the whole sequence? You start from a node and you do a random walk on the whole graph. You move from one node to another, and you just keep on adding the sequence after sequence after sequence and get the whole sequence. And the walk is a special type of walk known as Eulerian walk, where you visit one edge only once, because that's a constraint that we use in line with the algorithm that we are using. So generally, this is the mathematical basis for how genome assembly is done, because genome assembly is very important. Even if we have reference genomes, in a lot of cases what we do is, a particular region of the genome is very complicated, we do want to do the assembly ourselves. So we generate reads for that region of the genome, and we use an algorithm like this. There are a lot of softwares available for implementing algorithms like this, but I just wanted to give you a slight mathematical overview of how this is working, right? I think, that's all. Next I'll be discussing the assignment questions. We are running a little late, so if you have any questions, quickly. So let's let me go there. So the idea is that when you are you want to penalize where there is more overlap. So if the length of the overlap between two different nodes is very high, then what you use is basically the cost would be the negative length of the overlap, because what you want to do is you want to find how do I put it, like so in an algorithm, so if you have a very high overlap, right, between, say for example, node 1 and node 2, if you have a very high overlap, it's highly likely that these two nodes have originated from the same read, right? So these are not the set of overlaps that you are looking for, because if you already have got a high overlap, then what you are doing is you are taking the path of least resistance, right? So you're moving from point A to point B, but what you're doing is you're taking the path of least resistance. So that is the most amount of overlaps that are present between the set of nodes, but that is not what you want to do. You want to find the whole structure. So what if you do that, if you take the path of least resistance, what you're going to get is a very compact read which does not actually exist in the data. What you want to do is you want to find the smaller segments of overlaps that can explain much larger part of the genome compared to a smaller length of the genome but with very high overlaps. I'm not sure if I explained myself properly, but yeah, that is the idea. That you want to prioritize uh, the ones that are not the path of least resistance. I don't know if it's clear or not. Random walk will not give you. So that's the whole idea. So there is this whole. So you can prove it mathematically that so for random walk we were using. So we were the idea is that you go to a node and you calculate the probability of moving from this node to other nodes. So in the first iteration, as I was saying, you're in node 1. In the next iteration, 50% chance you can move to node 2, 50% chance you can move to node 3, right? And from here, node 3 you have 1, 2, 3, 4 options you can move, and so on and so forth. So basically the idea is that you calculate the probability at each step, and then you kind of sum up the probabilities for each iteration across different paths, and the path with the highest probability will tell you the best walk. So that's the thing, like it's like there is no guarantee that it will give you an accurate sequence. The idea is that the the sequence with the highest probability with respect to the random walk will give you the best possible outcome, is what we assume. Okay, any more questions? Yeah, just if you want to learn more, just look up De Bruijn graphs, look up Eulerian walks, look up, like, the first thing that you should know about is basically the traveling salesman problem, the what that I just skipped very conveniently, uh, but that is the basis of all the, yeah, the traveling salesman problem is that you can only visit one node single time. The corresponding graph theory algorithm for this is you can only visit one edge once, which is the Eulerian walk, right? So yeah, if you are interested, just look up the traveling salesman problem. You'll get a general idea about how walks and how graphs are kind of used in applications like these. Okay, any more questions? Okay, if not, I'll be going to the assignment questions. We do not have much time for the assignment questions, so I'll reduce the amount of, excuse me, I reduce the amount of time that I keep it on screen. So yeah, try to answer a little bit quickly. Yeah. Just 1 second. Yeah. The answer is basically SLR stands for synthetic long reads, firstly, right? So option B as A is definitely SLR stands for synthetic long reads. So across all three you can do. Just that you cannot do it in Chicago library because uh, they use partitioning and cross-linking to preserve this long-range information. It's not a synthetic method. It's a natural method. Rest of them are synthetic. So synthetic long reads are used everywhere across. Sorry, I just with respect to Sanger, in Sanger, other than Sanger, everywhere you can use and generate synthetic long reads. And nobody, I'm surprised nobody answered A because SLR stands for synthetic long reads. Okay. Okay, a couple of these. The answer is all. So, firstly, I would like to point out the first and the second option are the same. Right. So, emulsion PCR and gel bead emulsion is basically it's gel bead emulsion is the specific emulsion PCR that is used uh, for 10x, right? And yeah, so they are, after that the amplification is done using the amplification is done using the emulsion and the gelin bead emulsion PCRs. And first further the amplification is definitely done using the first two techniques, and pooling will also be done to create the library. So actually all of them, and first and the second options are exactly the same thing. So yeah, a lot of you answered B, but yeah, actually all of them is the answer. So one thing, when you see boxes like these, most of them are like multiple option correct, right? So generally, if you have circles, it will be single option. If you have squares, mostly it is going to be multiple option, but there also it can be single option. Okay. C and D is the correct answer. Splint circulation, I think I had mentioned very briefly about splint. Just let me check. Yeah, single stranded DNA circles using splint oligos. The splint circulation is the correct answer. Okay, just yeah, and adapter ligation, definitely. So yeah, C and D are the correct answers. Okay, do we not? Sarita, have you, like, this the answer for the current one or the previous one? Okay. Okay. Anyway, actually the C option is correct. So generally, SAM is sequence alignment format. SAM is the human readable version of the alignment file. From FASTQ, you after you do the alignment, the file that is generated is SAM file. We convert a SAM file to a BAM. BAM is basically the binary version of the SAM file. It's the same information is same is there. It's just in a much more compressed format and in a binary format. It's not in a human readable format. So BAM is smaller than SAM, and if you sort the SAM file, you can compress it further. So sorted BAM, sorry, if you sort the BAM file, you can compress it further. The sorted BAM file will be slightly smaller than the BAM file, and the index file is will be the smallest. It will be probably 1,000th the size of the actual BAM file. So yeah, moving on. Just give me a moment, I'll be back in a minute. Yeah, sorry for this. Okay, B and C mostly. Okay. Yeah. So for PacBio, the read length is kind of fixed in with respect to hi-fi and other long reads, right? The only USP of hi-fi is lower error rate or like higher accuracy. So that circularization that we talked about, that whole thing, that is the USP of hi-fi reads, that it is a circular DNA and it is like less prone to make errors. Not PCR is not getting used, DNA polymerase is getting used, all those things. The error rate is low. The read length is same for the technology across different types. For hi-fi, the only thing for hi-fi is basically the error rate. Okay, moving on. Okay. Okay. I think these are B, C, and D, if I'm not wrong. Yeah. Okay. So what the question is, disadvantage of synthetic long read technology, right? So when you're doing synthetic long reads, they rely a lot on computational assembly of these shorter fragments. So basically, these are not actually longer fragments that you have sequenced, right? These are shorter fragments you have computationally merged one on top of another with the help of a graph alignment that I just showed, and then you make a single long read out of that. But that technically is not a single long read. There are smaller reads, and uh, so if the barcodes are misread, or if two different DNA fragments get packaged in the same microreactor, then it forms a chimera. The software will incorrectly assemble them, and yeah, so barcode mismanagement and chimera formation definitely can happen. And also, since you're reconstructing, these are not like original long reads you have sequenced. You have sequenced shorter reads. You're trying to reconstruct it into a longer read. Ultra long reads are not possible. Reads greater than 50 kb are not possible, which is possible in PacBio or Nanopore, actual long read technologies. So B, C, D are the answers. Okay. Okay. The question is, which of them have controlled molecular scarring, meaning less molecular scarring? So sequencing by binding, that's its whole USP, that you won't have chemical remnants on a DNA strand after removing the fluorescent determinators. And mostly natural sequencing by synthesis, which is by Ultima Genomics. This technology is specifically designed to use natural nucleotides, minimizing chemical scars and improving subsequent kinetics. So A and B are the options. Definitely A and C are the options, right? So reverse transcription does not matter. Reversible transcription also, termination also, like you're going to have some scarring. Okay, moving on. Yeah. Okay, yeah, RCR is fine. Ligase chain reaction, yeah. I was also not aware of this. So this is the answer has been given by NPTEL only. Uh, yeah, LCR also is there. Yeah, ligase chain reaction. But rolling circular amplification is the main one. Yeah, I don't know. Sir might have mentioned LCR somewhere in the videos, but yeah, in NPTEL answers they have given it like this. So yeah. Okay. Yeah, we have not discussed high C, but yeah, I think sir has discussed high C, so should be fine. Okay, a few answers here. Okay, the answer is basically all. So, yeah, I have not discussed high C, but high C basically captures the chromosome in its conformation, right? If so, the idea is that if two DNA segments are, say, for example, 5 million base pairs away, generally from just a sequence, we cannot say okay, if those two segments are interacting or not. But due to packaging of the DNA with respect to the histones and all those things, euchromatin, heterochromatin, so these DNA segments which are millions of bases away, they can come in very close proximity. So to understand that, we do things like that. We do high C experiments. So the intact cells are cross-linked to freeze the 3D structure. The DNA is cut with a restriction enzyme. The cut ends are filled with biotin, which is the affinity labeling, and then joined together, which is proximity ligation. And then the DNA is sheared, and the biotin labeled junctions are purified via streptavidin pull down before sequencing. So yeah, all the steps are involved. Okay, moving on. It's a spatial proximity, basically. As I was saying, if you have if you have a DNA segment which is very far away from in terms of the sequence, but still those DNA segments can be in physical close proximity due to the packaging of the DNA. So what you are trying to see is physically what is the distance between different segments of DNA, different DNA fragments, and what it gives you is basically a contact map, which is a heat map, and it shows you topologically associated domains and loop formations and so on and so forth. So basically, yeah, for high C, will tell you the spatial proximity. Like even if sequence is very far away, the spatial distance might not be, and they might be interacting with each other. So that is why high C is used. Okay, moving on. Okay. Yeah, the answer is all. So this is not something we have discussed today, but it is discussed in the recorded lectures. So yeah, so this Chicago method is basically developed by Dovetail Genomics. It captures long-range genomic interactions in vitro. High molecular weight DNA is extracted and reconstituted in the chromatin in artificially adding in nucleosomes. Then it is cross-linked, cut, biotin-labeled, ligated, and then the crosslinks are broken, and then sequencing is done on the DNA. So all the 4 steps. this I have told too many times in today's session. This should be easy. Okay. No one says it. Okay, I'll wait 30 seconds more. Yeah, I think one of you gave a correct answer. Other than the accuracy, everything is better in long read. SV detection, the length of the SV, yeah, very well. Haplotypes, phasing is basically which of the variants are coming from the which of the parental strand. So for the mother strand, which of the variants are coming from the father strand, which of the variants are coming. So that is what phasing is basically. So if you have a longer read, it becomes much easier to phase. For assembly also, if you have a longer read, it becomes much easier to do the assembly, and obviously longer reads can help detect the size of the larger variations easier. Okay. Okay. Great. Hmm. Yeah, the correct option is B. So basically, use a tool called SAMTools. SAMTools can manipulate SAM files, BAM files. It can convert between each of them. It can do the indexing and all those things. So when you do SAMTools sort, the default behavior is coordinate sorting, and it is first sorted by the config name and then by the coordinate where the alignment begins. So yeah, that's correct. Yeah, this should be very easy. Yeah, that's correct. It's sequence alignment map. That is what SAM stands for. And if you this is a human readable format. If you convert it into binary based format, that will be BAM, and this is SAM. So yeah, moving on. NPD. Okay, different answers. Okay, the answer is higher read length and higher accuracy. So that would be A and C. Okay. Uh, okay. I am not very sure about. Uh, just give me a moment. Let me check one thing. Yeah. Okay. So yeah, if it would have been packed bio hi-fi, the accuracy would have been much higher for hi-fi compared to nanopore. But yeah, if you are using nanopore over CLR, continuous long reads, then the accuracy of nanopore is higher, and nanopore in general has a higher read length compared to packed bio. So that is definitely there. It is not going to PacBio is not going to produce higher longer reads than nanopore, but if you do hi-fi PacBio hi-fi, then definitely the accuracy is much higher compared to long read ONT. But here you are using CLR. CLR has lower accuracy than ONT. So yeah. Yeah, I'm not going to wait here. So full form of MNSBS is mostly natural sequencing by synthesis. It uses unmodified nucleotides mixed with a small fraction of fluorescently labeled terminators. So, yeah, it's Ultima Genomics. I think somebody answered it correctly. They use MNSBS. So, okay. Yeah, it's the first two options. It so VCF file. I think I can show you next day. Yeah. So VCF stands for variant call format, right? So VCF you find variant calls for populations, for individual samples, whatever, however you want to design the VCF file. It can be used to find SNPs. Even you can store larger variations there as well. Just it is the third option is very bad. Like, it is used across all clinical settings and also research, population genetics, etc. All of them it is stored. Uh, VCFs are used. VCF does not store raw data. VCF stores variant called data, like which position, what variant is there. That information is stored in VCF, not any kind of raw data. So A and B are the correct answers. That's correct. Okay. Yeah. Moving on. Yeah, the answer is four corresponding to the four DNA bases, A, C, and G and T. So yeah, that's correct. Okay, final question. Yeah, this is very easy. Yeah, that's correct. Single nucleotide polymorphism is what it's called. Okay, yeah, so that's all from my side. If you have anything to ask, do ask. Else we'll close the session. Oh, see, I do not like generate these questions by myself. These are like older assignment questions on the same course only. So I have to discuss these questions only. If you want anything extra, I can try to do that. But these questions I definitely have to discuss. That is in my mandate. So, yeah. I think in the upcoming sessions there will be problem questions. So, yeah. Okay. And one more thing I wanted to address is, see, I'm a PhD student. I'm not a professional teacher or a professor or anything like that, and I do not have experiences of teaching courses. Like I have taught NPTEL courses like this earlier, but like this is not exactly like completely teaching everything, right? So this I am covering whatever has been covered in the this week's sessions and all those things. So if you have any suggestions on how to approach for me on how to approach these sessions, or if you want something more, something less, something different, please let me know. And if you have any questions or anything, please stop me while in the middle where I'm speaking or not. Do stop me and ask me questions. So that is not a problem. And if you have any suggestions, please do let me know. So I can obviously try to improve the sessions. Okay. Anything else? Okay. Thank you guys. I'll close this session then. We'll see you next. Real data questions also help a lot. Yeah, I understand. I understand. Yeah, but the thing is, whatever, like, so these are the set of 20. So every week I have a set of 20 questions I definitely need to discuss. So what we can do is, so at the end of the course, so what happens is, this is I think 8 week course. So after 8 weeks, there will be one extra session where I'll be compiling all the information that I'll have been discussing for the 8 weeks. So there, if you have anything like extra for me to discuss or extra questions, I can try to find out. But in this session, I definitely have to try to discuss those 20. And if within that there are data questions and numericals and other things, I'll definitely discuss those. Outside of that, I do not make those questions. So if you want, I can make, like, maybe compile all of these things into the last session and discuss something like that, or like, yeah. Let me know if you want to have. Yeah. Okay. I do not have any comparison slide like that. Uh, I think things like this can, like Claude, ChatGPT, and not ChatGPT, I would not trust that much, but Claude or Gemini, probably you can just give in this prompt, they'll give you a uh, like full comparison. I personally do not kind of have that, but yeah, I can definitely make one. So yeah, yeah, okay, I'll keep that in mind. Okay, anything else? Okay, guys, thank you for joining. We'll see you next week. Bye bye.